Welcome to MoMed, where molecular medicine meets the future of healthcare. Together, we'll explore the fascinating world of genetics, molecular biology, biotechnology, and precision medicine through clear, evidence-based, and easy-to-understand explanations. If you're passionate about medical science, don't forget to subscribe, give this video a like, and share it with others who love learning. And turn on the notification bell so you never miss our new episodes, released every Sunday and Wednesday. Now, let's begin. Welcome to today's presentation on next-generation sequencing, commonly known as NGS. Just over 20 years ago, sequencing the first human genome required more than 13 years and cost billions of dollars. Today, the same task can be completed in just a few days for less than $1,000. Next-generation sequencing reads millions of DNA fragments simultaneously, allowing scientists to identify genetic variations with remarkable speed and accuracy. This technology is transforming disease diagnosis, advancing research, and making personalized medicine a reality.
Every NGS experiment follows a carefully designed workflow. First, genetic material is extracted. DNA is then prepared for sequencing. Millions of DNA fragments are read simultaneously inside the sequencing instrument. Finally, powerful bioinformatics software transforms raw sequencing data into meaningful clinical information. From the laboratory to the computer, and finally, to patient care.
Every sequencing experiment begins with a biological sample. Blood, saliva, or tissue can all serve as valuable sources of DNA. Before sequencing begins, scientists carefully evaluate DNA quality and purity. Instruments such as the nanodrop spectrophotometer measure DNA concentration and purity. An A260 over A280 ratio close to 1.8 indicates high-quality DNA suitable for sequencing. Human DNA molecules are extremely long. Sequencing instruments can't efficiently read DNA at its full length. Scientists first cut DNA into thousands of small fragments. These fragments are repaired and carefully prepared to create what is called a sequencing library. Think of it as organizing thousands of pages before scanning an enormous book. Each DNA fragment receives a pair of special adapters. These adapters allow the fragments to attach securely to the sequencing platform. Unique DNA barcodes are also added. These molecular labels identify each patient's sample, making it possible to sequence many samples together while keeping every result correctly assigned. Most modern sequencing systems use a method called sequencing by synthesis. One DNA letter is added at a time. Each nucleotide carries a unique fluorescent color. Cameras capture every flash of light. The instrument then converts those light signals into the DNA sequence, letter by letter. The first output of sequencing is a FASTQ file. It contains millions of DNA reads, together with quality scores. One of the most important indicators is the Q30 score. A Q30 value represents approximately 99.9% sequencing accuracy, equivalent to roughly one error in every 1,000 DNA bases. The next challenge is determining where every DNA fragment belongs. Scientists compare millions of sequencing reads against the human reference genome. This process is known as alignment. Imagine assembling a giant jigsaw puzzle where every DNA fragment must fit perfectly into its correct position. Once alignment is complete, specialized software searches for genetic differences. These may include single nucleotide changes, insertions, or deletions. every detected variant is recorded in a variant call format, or VCF, providing the foundation for clinical interpretation.
Finding a genetic variant does not automatically mean disease. Clinical experts evaluate each variant using internationally recognized guidelines, including the ACMG classification system. Variants may be classified as pathogenic, benign, or of uncertain significance. This final interpretation transforms complex genomic data into practical medical decisions. From diagnosing rare diseases to guiding cancer treatment, supporting reproductive genetics, and enabling precision medicine, next-generation sequencing is reshaping the future of healthcare, one DNA letter at a time.
Thank you for watching. If you enjoyed this video and would like to learn more about molecular medicine, medical genetics, and the latest advances in biotechnology, please support the channel by subscribing, leaving a like, and turning on the notification bell so you never miss our latest educational videos. New videos are released every Sunday and Wednesday. See you in the next episode here on MoMed.